So homopolysaccharides, it is consist of same monomer, that is starch and glycogen, and heteropolysaccharides, they consist of different monomers, example hyaluronic acid. So this is the difference between homopolysaccharides and heteropolysaccharides. Homopolysaccharides also called as homoglycans and heteropolysaccharides also called as heteroglycans. Homo means same, hetero means different. So that is the major difference between these two. And it is homopolysaccharides composed of single type of monomer, whereas heteropolysaccharide composed of different types of monomer. And this homopolysaccharide, as I said before, it is having the same repeating units of sugar molecule, and a heteropolysaccharide different repeating units of polysaccharides. And single type of monosaccharide is involved in this formation and different types of monosaccharides involved, and this is having the simple structure and this is having the complex structure. And the examples we already seen: starch, cellulose and glycogen, pectin, and this examples are having hyaluronic acid and chondroitin. So coming to the polysaccharides, we have seen starch, cellulose and glycogen. In starch there are mainly two components. One is amylose and another one is amylopectin. So amylose and amylopectin, it is a form of starch component, and the major difference between amylose and amylopectin is amylose is a straight chain polymer, that is it is present in the linear chain format, and amylopectin, it is appeared as branched chain form. So this is the major difference. So it is a straight chain polymer of D-glucose units, and it mainly constitutes about only 20% of the starch, and in amylopectin it constitutes about 80% of the starch. Amylose linear, amylopectin branch. And coming to the properties of carbohydrates, we may come across through physical properties and chemical properties. And in physical properties, first one is stereoisomerism. So along with the structure, the carbohydrates have two properties, and stereoisomerism is nothing but it is having the same formula but different spatial configuration. For example, D-glucose and L-glucose. D-glucose and L-glucose having the same formula of C6H12O6, but they only differ in the spatial configuration, that is from right to left. So you could see the left side example of D-glucose and L-glucose. D-glucose HOH, HH all are in the left side, whereas the mirror image of the D-glucose is L-glucose. So whatever is in the left side and it goes to the right side of L-glucose. So only the spatial configuration will vary, and otherwise the structure and the molecular formula are all same. And next one is diastereoisomers, and it is having the configurational changes only at the second carbon, third carbon or fourth carbon. So you could see the right side examples of mannose and galactose, where you could see D-glucose which is in the center. In mannose, the D-glucose second carbon will change accordingly as like mirror image, and in D-galactose the fourth carbon will change the fourth carbon as mirror image in the galactose. So this is the stereoisomerism and diastereoisomers structural differences. And coming to the chemical properties, first one is oxidation. And oxidation, it is nothing but the addition of carbon to the glucose. So you could see the example of in the left side. So D-glucose, when oxidized, it produces D-gluconic acid. So that is the structure. We want to write D-glucose in the left side, and only the oxygen atom is added to the D-gluconic acid, and this oxygen atom is added only in the first carbon. So in D-glucose we have aldehyde functional group. In gluconic acid we are having the carboxylic group, that is COOH group with the first carbon. And second one, reduction, and this carbonyl group reduce into alcohols. So CHO is the aldehyde group and it is reduced to alcohol OH group. So you could see the second side example, D-glucose by using the sodium borohydrate, it gets reduced into alcohol that names the D-glucitol, and this is the aldehyde group. So this carbonyl group reduced into alcohol, which is often termed as alditols, using some of the chemicals or metals, and the aldehyde form converted into alcoholic form. That is why it is named as such.